I mean, here we go. Path Grand
All right.
Round.
Thanks, Larry. So welcome everybody to another edition of the Grand Rounds. It's my great pleasure to introduce Caitlin Latimer, who most of you know, so she doesn't need much of an introduction, but I will anyway. She was summa cum laude and a D1 full scholarship athlete at Vanderbilt, did her MD PhD at University of Kentucky, and set all kinds of new standards with our friends out there. But has really made her impact since she's been here at UW. She did her anatomic pathology residency and neuropath fellowship here. Looking at her CV is just full of awards. I want to highlight just a couple. She got she was the recipient of the Moore Award in 2018 for the best clinical pathological study for our annual neuropath meeting. That's given out once a year and rarely to a junior person. And so that was a big deal. And then, of course, most of you know she is the inaugural recipient of one of the two Brown fellowships.
Great, I got the 2 bullets.
Fantastic. And also just got a Building Bridges Award.
Okay, wait,
she has funded her faculty development the whole way with a competitive career development award through the Alzheimer Center Network, and then with a K08 and a bunch of other funding along the way. I count 18 published peer-reviewed manuscripts since she's been with us here at UW, and she's going to tell you a little bit about the work that she's doing in her K, and is really turning a lot of heads in our research community. Her talk is entitled "Resistance and Resilience to Alzheimer's Disease, Pathology in a Community-Based Cohort," and I'm looking forward to hearing about this. So take it away, Caitlin.
Great. Well, thank you, Dirk, for that introduction, and thank you, Larry, for the opportunity to present here. So some of this work is going to be stuff that I've talked about in various places, but this is my first time getting to present at Path Grand Rounds. So I'm going to share my screen, hopefully. And is that working? All right.
Yes, it is.
Great. Okay. So like Dirk said, I'm going to talk about resistance and resilience to Alzheimer's disease pathology. And this is a topic that's really been a major focus of my research for a while. And I spend a lot of time looking at neuropathology in a variety of cases from individuals both with and without dementia. And what I've really come to appreciate in doing this is how much this observational pathology can really inform our understanding of neurodegenerative disease processes. At least it can open our eyes to potential new avenues that we need to pursue in our research in order to find some therapeutic strategies for these terrible neurodegenerative diseases. So to that end, I have a few kind of major takeaways that I hope you guys will come away with. And the first is really just how I'm defining resistance and resilience to Alzheimer's disease. And on the surface, these terms may not seem all that controversial, but it really depends who you're talking to and how you want to define those. And so what you're getting today is sort of a neuropathologist perspective on resistance and resilience to AD. I also want to hope you'll come away with this idea, although I think a lot of people here already know this. Not everything that is dementia, especially in the elderly, is plaques and tangles. And so I'm going to particularly highlight not a new pathologic protein, but sort of a new way of looking at it in the neurodegenerative field. And then finally, I will get into a little bit of my K research, as Dirk alluded to, and specifically what I hope you'll take from that is how I think pathology really plays an important role at that intersection between the basic scientists and generating models of human disease, because as pathologists, we're the ones seeing the pathology. And so I think it's really important that we're right there helping to inform these models, because the models are only going to be as good as their ability to recreate the pathology. So before getting into all that, just to kind of put us on the same page as far as Alzheimer's disease, I think most people appreciate it's a very common and very devastating neurodegenerative disease. It typically presents as a memory-predominant illness and then progresses to involve more severe and widespread cognitive decline. It is thought to be the underlying cause of dementia in 50 to 75% of cases. And although there are known genetic causes, those actually are quite rare, and by and large, the most common form of Alzheimer's disease is the sporadic late onset form. And unfortunately, those cases, the genetics and etiology is much less well understood. And so that's where a lot of the research is focused, is trying to understand sporadic late onset Alzheimer's disease. I also think this group is probably pretty comfortable with the pathologic hallmarks of Alzheimer's disease. So we have the amyloid plaques, which are these extracellular accumulations of amyloid protein, and the intracellular neurofibrillary tangles, which are composed of hyperphosphorylated tau. And then a third lesion is sort of a combination of the two that we look for, which is a type of amyloid plaque, the neuritic plaque, which also has some dystrophic tau-positive neurites sort of mixed in that highlights really well on a silver stain. So when reviewing cases for neurodegenerative disease, we look for all three of these types of lesions. But what's really vexing about these so-called pathognomonic lesions of Alzheimer's disease is we actually see this pathology in almost every aged brain, whether there was a history of dementia or not. And it's just more a question of how much pathology you see. So in 2012, the field came together to sort of come up with a staging scheme so that we could sort of group the pathology into, a sort of low, intermediate, or high Alzheimer's type of pathology. And that's mostly based on how many brain regions contain these pathologic lesions. And that is because a lot of the prior work had demonstrated that both amyloid and tau appear to progress throughout the brain in very stereotypical patterns, such that the further along that process the pathology gets, the more likely the individual was to have had dementia in life. So for amyloid pathology, we use Thal phase. Thal described amyloid as starting out in the cortex and then moving deeper into the brain. So the last place it goes is out into the posterior fossa and brainstem and cerebellum. Brock and Brock described the progression of neurofibrillary tangles as sort of an opposite progression to that of amyloid. It starts in the brainstem and moves into the hippocampus, and the last place we see it is in the cortex. But where these two staging schemes, kind of what they have in common is that neither of them really tells you anything about the burden of pathology in any of those given regions. It's just sort of where the pathology is. And so that's an area that I think research can really add a lot to as far as our understanding of these concepts of resistance and resilience, because you can have an individual with a high Brock stage, which means they have neocortical tangles, but how much pathology do they have, and is it important to try to tease that apart as well? So I think we all know that Alzheimer's disease is really common and devastating, and our chances of developing dementia increase as we age. But we also know that there are plenty of individuals that live well into advanced age and maintain their cognitive function remarkably well. So these are very interesting individuals. And on the surface, they all look very similar. It's an elderly person who is highly functional. But neuropathologically, we know that they come in actually two basic flavors. So you can have someone who's cognitively intact because they never actually developed the pathology necessary to cause cognitive issues. So these are the resistant folks. They have avoided pathology. And that's different from the resilient individuals who actually have developed pathology in their brain, but somehow their brain has been able to cope with that pathology and they're considered resilient to the pathology. Now, both of these groups are worth studying and are the topic of a lot of intense research, because we really think that if we can understand the mechanisms underlying resistance and resilience, it may serve as a roadmap to figuring out interventions and treatment strategies to help people who don't have those mechanisms already in place. So how do we go about studying people who are considered resistant and resilient to Alzheimer's disease? And I would argue that one really good way to do that is to really dive into the neuropath data of a well-characterized community-based cohort of individuals. And so here in Seattle, we have one such fantastic cohort. It's called the Adult Changes in Thought Study. And so for those of you who don't know, this is a population-based prospective cohort that is through a partnership with Kaiser Permanente, formerly Group Health Healthcare Systems. And what the study does is when individuals reach age 65 or older and are cognitively intact, they can enroll in the study. And it's really focused on brain aging and risk factors for dementia. So these these participants are really part of what the study calls a living laboratory. So they're well characterized as far as their medical histories, what medications they're taking, their physical activity, and every 2 years they undergo a cognitive assessment. And so that's just a screen to see how they're doing cognitively. And if their score drops below a certain point, then that reflexes to more intense neuropsychological testing so they can receive some sort of diagnosis for their dementia. And then ultimately, we want to study the brains of these individuals. And while the study itself doesn't require that an individual agree to donate their brain at the end of life to be a part of the study, we do get about 30% of participants to consent to autopsy. And so we have now somewhere between 850, 900 brains in our brain bank from this cohort. So in that autopsy cohort, we have a nice split of cases from cognitively intact individuals and those with dementia. So it's about 50-50. And what you can see in this figure down here is the nice spread of pathology that we have across both the non-demented and demented subjects. So each of these bars represents an individual in the study, and then the different colors are the different types of pathology that we look for. And so even in non-demented individuals, you see there's quite a range of pathologies that we see. So this is a nice cohort from which we can draw upon to develop a study on resistance and resilience to Alzheimer's disease. And so that's what we did. But first we had to come up with our definition for resistance and resilience, and that is a little bit tricky. So first you have to define the cognitive function, and you're always going to run into this question of whether you have a subject who's truly resilient or have they really just been hanging out in a preclinical phase. And if they had lived another 10 years, they would have become demented. And we can't really answer that, and it's really hard to know that, but because the individuals in this study tend to live to be about 88 years old, I think is the average age of death. And if they are deemed cognitively intact by the study and that they haven't had any real major issues with performing their daily tasks, and a lot of them are still living on their own, that really embodies, I think, the true definition of cognitive resilience. These are individuals worth studying for that reason. So to that end, we did try to pick as cognitively robust individuals as we could. So we did use a cutoff of individuals had to have a cognitive assessment within 2 years of death, and that just helps reduce the chance that we accidentally select someone who had converted to dementia that was missed in our resilient or resistant cohort. Then we have to define the pathology. Now, because Alzheimer's pathology is sort of on this continuum, and you have normal individuals with pathology, at really low levels, what truly just is resistant pathology versus resilience. And so there are a variety of ways we could do this, but we decided to go with the most extreme definition that we could and yet still have cases to study. So for the resistant group, that meant looking at cognitively intact individuals with a Braak stage of 3 or less, and that's out of 6 stages, and no neuritic plaques. Also, these individuals, we said, had to be 85 years of age or older, and that was just to help ensure that we didn't end up selecting for individuals who had died at a younger age before they had the opportunity to develop Alzheimer's disease pathology in their brain. So that ended up with about 14 individuals. In the resilience group, we used the highest stage of pathology that we can ascribe to Alzheimer's disease. So a Braak stage of 6 out of 6 and frequent neuritic plaques. And so then once we had our resistance and resilience cohorts, we matched them to individuals with a clinical diagnosis of dementia and pathologic high Alzheimer's disease. And our approach initially was just to perform traditional neuropathology. These cases were spanning the entire duration of the study. So some of them were from very early on, and we've changed how we look at neurodegenerative disease over the years, whether it just be changing antibodies, adding new diagnostic entities. And so we wanted to make sure we looked at everybody with the same tools, the same criteria, the same neuropathologist. And so we re-reviewed everything, not just for Alzheimer's pathology, but all the general neurodegenerative disease processes that we look for. And then to really go beyond that, we wanted to do some quantitative pathology. So we did apply some digital slide scanning and quantitative measures as well. So before I get into some of the findings from this study on resistance and resilience, what were some of the things that we thought we might find? So I mentioned we wanted to dive into the quantitation, and that one of the issues that we have with the diagnosis of Alzheimer's disease is that it really lacks those quantitative assessments. So could it be that individuals would meet criteria for high Alzheimer's pathology using traditional neuropath techniques, but if we actually looked at the burden of pathology, would we actually find that it was a lot less in the resilient subjects than in the cognitively impaired subjects? Also, it's pretty well accepted that as we age, we collect multiple types of neuropathologies in our brain, and those that are demented tend to have more than one as the rule. And so could it be that resilient subjects, while they have Alzheimer's pathology, maybe they lack other types of neurodegenerative disease? So it's really more resistance to other types of neurodegeneration. And then similarly, the resistance group would be a really interesting group to dig into this question of whether there's some sort of global resistance to neurodegenerative disease, such that if we could figure out that mechanism, maybe you could prevent multiple types of neurodegenerative processes because they're all really linked to some sort of pathway. And the resistance group would help us answer that if they were not only resistant to Alzheimer's disease, but the other neurodegenerative processes that we were looking for. So what do we find? I'm going to start with the resilience group because they had pathology, and looking at the presence of pathology is more interesting than those without, which was what defined the resistant group. And so I mentioned we were looking at disease burden in addition to the progression of pathology throughout the brain. And so here I have a couple micrographs, one of a representative resilient subject on top and an AD dementia subject on the bottom. And just by eye, and this is from the frontal cortex, you can see that there's not really a huge difference between the amount of pathology in either case. Now this is from the cortex, and even though we used a neuritic plaque score to select on cases, which is, looking specifically at that type of plaque, and this is really staining for all types of plaques, there is going to be a relationship. And if you have more neuritic plaques, you're going to have more total plaque pathology. So it's not all that surprising that in this group that was selected to have high neuritic plaques, we don't see a difference. And over here is my bar graph, which is just, again, a very semi-quantitative assessment of the severity of the AD pathology, and no difference across the cortex. Now, we didn't select these cases based on the Thal phase, which is the progression of amyloid through the brain, and that's because that's not a data point that we had on every single case, because it was something that got added later on. And so I did look to see if there was a difference in Thal phase, and it wasn't significantly different. But if you look at the amount of amyloid pathology in the higher Thal phases, specifically the midbrain and the cerebellum, then that's where it does start to look like there may be more amyloid burden in those regions. And so we did not do digital scanning and quantification in these regions or in this pathologic peptide, but that's something that I think we want to follow up with. And also, again, this was only 7 subjects, so a very small cohort of very specifically defined individuals. We want to follow this up with the greater ACT cohort. But that was really the design of this study, was to start to identify areas that we wanted to pursue further as far as leads on what really is resistance and resilience to Alzheimer's disease. And so I think this is an example of that. And so we did the same thing with tau pathology. We wanted to look at tau burden. And by design, all the cases met criteria for high Braak stage. So over here, we have a micrograph of a resilient subject and subject with dementia. And you can see up here, these are neurofibrillary tangles. So it does meet those criteria. But what I hope you also notice is that this demented subject has a lot more staining. And so this is an antibody to phosphorylated pathologic tau, and there's just a lot more of it in the cortex in the subjects with dementia than those that are resilient. And so when we looked at this across multiple brain regions, and this is again just my semi-quantitative assessment, where the significance jumped out was in the frontal cortex, which is one of the later stages, but also a stage where we would really expect the pathology to have an effect on cognition, or at least global cognition. And we did follow this up with digital slide scanning and quantification for the staining on the slide. And it just confirmed what we were seeing by eye, that there is more tau in cases with dementia. Even though these cases do meet that criteria for AD, there's just a lot less burden of tau pathology. And so going back to those hypotheses, I do think we sort of proved that theory correct, that there is less tau burden, less amyloid burden in specific brain regions that correlates with resilience. So perhaps it's not so much that you're resilient to the pathology, but you're resistant to generating the burden necessary to cause cognitive issues. Then for these other two hypotheses, we need to look beyond the Alzheimer's pathology and into some of the comorbid pathologies that we see in aged brains. And so that includes vascular brain injury. So vascular disease is often comorbid with AD and commonly found in the elderly. And so we're looking at infarcts, like strokes, and microinfarcts, which is shown here. You can only see them microscopically as sort of a little glial scar. And then also looking at vessel disease in general, atherosclerosis and arteriolosclerosis. We looked at Lewy body disease. Lewy bodies are alpha-synuclein positive inclusions found in certain subsets of cells throughout the brain associated with Parkinson's disease. And then we looked for TDP-43 pathology. So TDP-43 is a nuclear protein, and it aggregates and is phosphorylated, and you can find those phosphorylated inclusions in the amygdala, hippocampus, and cortex. So these were the main things we were looking for. And just like in AD, where you can sort of give a severity score of like a mild, intermediate, and severe, we can do the same thing with vascular brain injury, Lewy body disease, and TDP-43 pathology. For example, brainstem Lewy bodies are not considered that high risk for causing cognitive issues, more the movement disorder of Parkinson's disease. So that would be sort of a low severity score. Limbic, now the Lewy bodies have moved into cortical regions and may have an effect on cognitive dysfunction. So that's a more intermediate risk. And then neocortical is really what's associated with cognitive dysfunction. And so giving each of these a severity score and then adding them together for each individual, we get a global picture of the amount of pathology each subject in the study has. And so this figure shows what that looks like graphically. So each bar graph is an individual, and then each color represents a different type of pathology. And so now we're looking at the resistant subjects, not the resilient, and you can see based on how they were selected, it makes sense that they have very low Alzheimer's pathology, which is this gray colored bar graph, compared to the matched AD dementia subjects, which have as high of Alzheimer's pathology as you can get. What was somewhat surprising to me was that there actually wasn't a big difference between the two groups in vascular brain injury, which is shown in the dark gray, and Lewy body disease in the black color. So there's sort of a smattering of cases across both groups. It wasn't significantly different. But what jumps out as being sort of selectively different is the TDP-43 pathology. So it was really quite rare to see TDP-43 pathology in resistant cases, but it was the opposite in the dementia cases. Almost every single individual had some TDP-43 pathology. And so what struck me about that is there almost seems to be a link between having Alzheimer's disease pathology and TDP-43 pathology. Similarly, we looked at the summary neuropath score in the resilient cases. And again, because of how we selected them, they have high Alzheimer's pathology. And again, when we looked at it statistically, there was the main difference between the AD dementia subjects and the resilient subjects was, again, TDP-43 pathology. So nearly every subject who was demented had TDP-43 pathology, and hardly anyone in the resilient group had it. So the takeaway that I took from this is TDP-43 pathology is really associating with dementia. And so if we go back to those hypotheses, resilience to Alzheimer's disease may somehow be related specifically to resistance to TDP-43 pathology. And what I also found interesting is that if you're resistant to Alzheimer's disease pathology, you also seem to be resistant to TDP-43. And so that suggested that there was some kind of relationship between Alzheimer's pathology and TDP that is really hard to answer, to really look further for in autopsy tissue, because now we're starting to ask questions about this relationship and is it dynamic. But I guess the first question to answer is, what is TDP-43? And the first thought I had was, is this just a marker of death and destruction? And it's showing up as concomitant with the AD pathology and dementia because it just shows up when everything is dying. And again, that's hard to know in autopsy tissue because it's one time point and it's observational. But the opposing theory would be that TDP-43 pathology is actually actively contributing to the neurodegenerative process. And based on what we know about TDP-43 pathology, I don't think it's really that out there to lean towards that second theory. And so I wanted to spend a little bit of time going over TDP-43, what it is, what it does, and then kind of talk a little bit about how it sort of made its way onto center stage in neurodegenerative diseases recently. So TDP is transactive response DNA binding protein 43. It's encoded on chromosome 1, and it's expressed in the majority of cells, and under normal conditions, it'll express in the nucleus. And there it's believed to be involved in a lot of really important processes involved in protein production. And specifically in the brain and nervous system, it's thought to play roles in important processes like mitochondrial metabolism and cellular stress response and synaptic integrity. And so these are obviously important things involved in neurodegenerative diseases. And the reason we know so much about TDP-43 is because of its relationship with other neurodegenerative diseases. So in 2006, I believe, TDP-43 pathology was identified as the major pathologic protein associated with inclusions in the neurodegenerative disease, amyotrophic lateral sclerosis. So this is a neurodegenerative disorder that leads to destruction of motor neurons. So what they found is that there were these ubiquitin positive inclusions in motor neurons, both in primary motor strip and in the anterior horn cells. And in 2006, those ubiquitin positive inclusions were found to mostly be made up of pathologic TDP-43. And then shortly after, around the same time, it was also identified as one of the major pathologic proteins of frontotemporal lobar dementia. So FTD is a clinical syndrome defined by kind of bizarre behavioral symptoms, a progressive neurodegenerative disease, and it can be caused by a number of misfolded proteins, but over half the cases are associated with TDP-43 pathology. And the reason I think that we consider TDP-43 pathology to be a causative player in these diseases and not just an innocent bystander is very similar to the Alzheimer's story. So we found a mutation in the APP gene, and so amyloid pathology, amyloid is thought to be a player in Alzheimer's disease. Similarly, the TDP-43 gene mutations were found and were linked to ALS and FTLD. So likely TDP-43 does play a role in the neurodegenerative process. Plus, given its important role in these cellular processes that would be essential to neuron function, whether it's through a loss of its normal function or gain of toxic function, TDP-43 certainly has ample opportunity to wreak havoc if something goes wrong. So when we talk about TDP-43 pathology, what do we mean exactly? So I said it's expressed in most nuclei throughout the body. So if you do an immunostain just to TDP-43, it should light up the nuclei under normal conditions. Through various studies in cell culture and other experimental systems, it's been shown that what likely happens first in TDP pathology is you lose that nuclear you lose the nuclear localization of TDP-43, and then it starts to accumulate both in the nucleus and in the cytoplasm in sort of these aggregates, which are associated with phosphorylation of TDP-43. So it's getting phosphorylated, abnormally folded, and aggregating. And so you can use either a stain for the non-phosphorylated TDP, which will show you those very early changes, or you can stain for phosphorylated TDP, which will just highlight the aggregates themselves. And as you can see just from this image, TDP-43 pathology comes in a lot of different patterns, and there's been a lot of work done on describing all these different patterns, and neuropathologists love to split it out as many ways as they can. I'm not going to get into that, but one of the things I think that happened after this was described is neuropathologists started looking for TDP, and the more you look for something, the more you find it. And over time, really over the last 10 years, there's been this steady stream of publications describing TDP-43 and other age-related neurodegenerative disorders. And what seemed to really come out was how often people were seeing it in association with Alzheimer's disease. And that when they were seeing it with Alzheimer's disease, it was correlating with really severe clinical features. The subjects had more rapid cognitive decline, more severe atrophy in structures like the hippocampus, amygdala, and temporal lobe. And so I'll admit I didn't know all of this about TDP-43. I knew it was an ALS and FTLD, and then I saw this independently in our ACT cohort. And when I looked to the literature to see if anyone else had reported this, I was really kind of pleasantly surprised to see that several groups had been describing this, but they'd all been doing it kind of off in their own little silos and using different terminology and different staging strategies, but they were all basically saying the same thing, that TDP-43 pathology is important in aging brains, and in particular in association with Alzheimer's disease. So while I was kind of grappling with all this and trying to figure out where my research fit in, Dr. Keene was actually off meeting with a bunch of experts in the field on this topic to do just that, to really try to bring the field together and give this entity a name and give pathologists a sort of unified approach to working these cases up and describing them so that it could hopefully lead to a more unified effort for studying this pathology. So the name they came up with was Limbic-predominant age-related TDP-43 encephalopathy, or LATE. And I remember when this paper came out, it caused kind of a big stir, and the media was calling it a new neurodegenerative disease, and it really isn't new. Like I said, it's been described by a lot of these people on this author list for the last 10 years, but it was really the first time it had all been put together and used a common language. And so one of the things they did was try to give us a way to work up these cases and screen for this disease process, very similar to how they did for Alzheimer's disease back in 2012. And so just like Alzheimer's disease with amyloid and tau, it looks like TDP-43 is sort of progressing stereotypically throughout the brain. And the pattern that it has is it starts in the amygdala, which is this purple blob here. It's a mesial temporal structure, sits sort of right in front of the hippocampus. And when it's found in the amygdala only, there aren't great correlations with cognitive dysfunction. So we're not really sure what it means when it's in the amygdala. But as it progresses out into the entorhinal cortex and then out into the hippocampus, then the study starts showing an association with episodic memory. And so that makes sense. The hippocampus is where we sort of consolidate memories. And then finally, it looks like it goes out into the neocortex in the final stages, and then that's when it starts associating with more severe cognitive dysfunction. And so I think this paper really highlighted two really critical points. The first was now we have a way to commonly describe and work up these cases. And it also brought to light the fact that there is quite remarkable overlap, both in the pathologic distribution of this protein and in the clinical presentation with Alzheimer's disease. And it is quite likely that there are individuals and were individuals walking around with, who presented with episodic memory complaints and progressed to more severe cognitive decline and were given the diagnosis of Alzheimer's disease. And maybe those individuals participated in GWAS studies or were part of clinical trials for Alzheimer's disease. And it's very likely they also did have Alzheimer's disease pathology. So even if you use your biomarkers for AD and diagnose them as having AD, TDP-43 pathology or late pathology is so common with that. We don't know who also has this pathology and how that might be confounding what we know about Alzheimer's disease pathology. So I think there's a lot of work still to be done in this area, but it was really great that the field finally came together and gave this a name and a working strategy to study it. And so I'm just going to kind of finish up going through some of the ways that I think our neuropath core and my specific interests are really aligned with this focus on TDP-43 pathology and Alzheimer's disease. So our resistance and resilience study was really quite small. It was 14 resistant and 7 resilient. So we want to make sure we look at the entire ACT cohort, which, like I said, is now reaching 900 cases. And because we haven't been assessing for TDP-43 routinely for very long, we only have about 183 of those cases currently worked up according to those consensus criteria. Now, the good news is we're seeing the same kind of trends, that TDP-43 pathology is more common when you have high Alzheimer's pathology. It's also more common in individuals with dementia than without. But there's still a lot of work to do. We have to finish characterizing the cohort, and that will include quantification of the pathology, because I think we need to go beyond just the staging. And this includes the TDP pathology, but also the Alzheimer's pathology. And so I just wanted to shout out to the True Brown Fellowship that's going to help fund some of that slide scanning and digital slide analysis, which I think will be really important to better understanding these relationships between Alzheimer's disease and late. And then finally, the observational neuropath is great. And to me, it's really brought up a whole host of interesting directions to look for potentially novel therapeutic targets and different pathways, but it's observational. And so we need a way to really get at that underlying relationship and look at the pathways of proteotoxicity. And so that's where these model systems really come in. And so I want to spend the last little bit talking about my new favorite model system, which is C. elegans. And so yes, it's a worm, and Alzheimer's disease is a uniquely human disease. So it seems like a great divide, but this is where I think pathologists are so important, because we can really help inform the models and work together with basic scientists to make these models as good as they can be. And so what's really neat about C. elegans is, well, first of all, it has a rapid life cycle. So you don't have to wait 100 years to study its nervous system. The other thing that's cool is they are transparent. You can study their nervous system while they're still alive. Even though they don't have a brain, they do have a very well differentiated nervous system. It's been completely mapped out, so they know which neurons talk to which other neurons, where they came from developmentally, and their genome was the first to be genetically mapped out. And there's a lot of homology with the human genome. And so you have now all of a sudden this enormous toolbox of genetic tools and molecular biology tools to really manipulate this genome and try to understand some of the genetic modifiers of the pathologic phenotypes. So what I was really interested in is the fact that there are quite robust models of tau and TDP-43 proteinopathy and C. elegans that have been used for years to study tauopathy and TDP-43 proteinopathy in isolation. And so I was intrigued by this, but I also wondered, is there a way we can study both pathologies together? Because that's what I'm seeing in the human brains. They're not separate, they're together. And I want to know, are they interacting? Is one doing something to the other? And so that's when I started working with my collaborators and mentors at the VA, Drs. Nikki Liachko and Brian Kramer, who have been working really successfully with these models. And they said, of course we can do that. So basically you can take a C. elegans that is expressing human TDP-43 and cross it with one that is expressing tau and create a double tau TDP model. And the really cool thing was when we did this, it actually recapitulated what I was seeing in the human brains. And that is, if you have just TDP or just tau, you have a mild effect on the normal phenotype. So they use movement assays to kind of assess how the nervous system is functioning in these organisms. And they're really not all that affected if it's just one or the other. Similarly, they don't have as much pathologic protein if there's just one or the other. So it's not really bothering the organism. But then when you cross them together and now you have both in there, all of a sudden the phenotype is drastically affected. These animals are very sick. They don't function very well. And all of a sudden you have all this TDP43 and tau, both total and phosphorylated forms. And so this I just found to be a really interesting way to start to probe some of the mechanisms and underlying pathways of these proteotoxicities. So this slide, I'm not going to go through details. It's just sort of a summary slide of how I see this really nice integration with pathology using our quantitative slide scanning and these digital analysis techniques, selecting out specific individuals, specific brain regions, and doing genetics, and from that identifying potential genetic modifiers, which now we have a really nice model to go in and test those genetic modifiers. And so that is sort of where my K work is going. I'm learning how to work in this model system, but it really all goes back to the pathology and how the pathology is informing the model. And then hopefully the model can come back and inform something about the disease process in humans. And so I think if I have a little bit of extra time, I did just want to end on a human note and also give a shout out to our remarkable neuropathology core and the work that they're doing. So one of the future directions that I think will be really important is being able to diagnose these individuals in life. We currently don't have biomarkers for people with TDP43. So even if we can identify all of the pathways and come up with potential therapeutic targets, we still need to figure out who these people are that have TDP43 pathology. And so one of the ways that this might be possible is through structural antemortem imaging. So we know that there are specific brain regions that seem to be affected by TDP43. And so is there a way to tease apart atrophy patterns in those with TDP43 pathology, those with AD, and potentially those with a mix? That might be asking a lot, but I figure we gotta start somewhere. And so now, thanks to Dirk Keen and our amazing autopsy technicians and Christine McDonald, who's a neuroimager extraordinaire, we get ex vivo postmortem MRI on all of our research brains. And they're able to perform this automated segmentation of the various structures within the brain that allows us to get volumetric measurements that are just not possible using traditional neuropathologic assessments of the brain. So right now, if I just were to assess atrophy in the brain, it boils down to me just saying mild, moderate, severe global atrophy. And if you use all of the tools that neuroimagers have at their disposal, you can all of a sudden break it down into all the different regions of the cortex and the subcortical regions and get actual volumetric data out of that. And so this is very, very early days for this, but I just wanted to get a sense of if this was going to give us any useful information as far as teasing out some of the differences. And so I just took all the cases we had that I knew had TDP43 pathology and those that didn't, and looked to see if there were any volumetric differences across the brain. And what we found is that the areas that were significantly different are those that also are known to be more affected by TDP43 pathology. So places like the hippocampus and the temporal lobe is significantly smaller in individuals with TDP43 pathology. So there's obviously a lot of work to do here. We haven't done any covariate analysis for all the other variables at work in these brains, but it's just a really neat direction that I'm excited about as far as sort of the integration with neuroimaging tools. And so with that, I just wanted to thank the village of people that make all this work possible and really fun, and of course the funding sources. And then I would be remiss if I didn't also thank all of our participants. So the donors and their families. If they did not provide us with this ultimate gift to science, none of this work would be possible. So, and with that, I'm happy to try to answer any questions.
Caitlin, that's terrific. Thanks so much. And we began a chat line. So let me just begin reading some of the questions. You know, I should have done that a while ago, but boy, I'm a slow learner on Zoom. Chat, there are quite a number of questions. And so let me just go to some of the earlier ones here. Let's see, first one here. Are TDP P43 mutations acquired with age, or are they inherited germline?
So the ones that I know of are inherited mutations. I believe they're familial or sporadic, but the TDP pathology of aging, we don't know of any mutations associated with that. So, yeah.
Okay. And here's a question. How were resistant and resilient cases matched with AD controls from the ACT cohort? Were you able to adjust for cognitive reserve, i.e. education or occupation?
So we actually, I didn't get into that whole side of things. We actually didn't select based on that because we wanted to see if that was a whole other place that would influence this idea of resistance and resilience. Was it to do with their education? And I don't believe anything came out significant when we looked at that. So there might have been a trend or something for education, but yeah, nothing came out of that analysis.
Okay. Let's see, next question from George Martin. Want to know if you could show early slides, the amyloid foci and controls and resilient. And he saw, and I seem to remember that while the number of foci did not appear to differ, the intensity of staining seemed to be higher in the controls.
Yeah, so I can go back to that, or can I can do that on here. And I mean, we did, I think it really depends who these were when they were stained. I don't think we restained if we were using the same antibody at the time. So it could have something to do with the staining. Using the intensity of an immunostain, I find to be really challenging.
Sure, that's always a big challenge. And actually, I remember looking at this also, and the dementia one seemed to have larger clumps than the resilient.
And so this is something I've thought about, not specifically that, but we are moving towards this digital slide scanning and using different machine learning approaches. And so would there be a way to start teasing out, you know, sizes of plaques or parts of the brain where the plaques are, their patterns, all that kind of stuff, I think could potentially be really informative. So moving to a digital approach, I think will add a lot of information to what we know about these diseases.
Yeah. So, Larry, can I just add to that? George, that's a great question. You know, these were selected as representative. Who knows how representative? I mean, you know, but we, you know, Caitlin didn't mention, but another effort that's happening in the Neuropath core is very complementary to this kind of immunohistochemistry, which, as you know, it's the staining intensity isn't a very linear function, and so fluorescence staining would be much better at assessing intensity, all done at the same time with the same conditions. Or people in our group have developed a tissue extraction-based protocol, kind of like what you would do with a Western, except with formalin-fixed paraffin-embedded tissue, using a Luminex platform as an immunoassay to quantify the proteins in an absolute fashion. Excuse me, some of that was done for this. I can't remember what the data looked like, but that would be I think, much better way to get to really overall burden, right? Because the digital image analysis that was done on this was just frequency and percent area. And those didn't come up different, if I remember, Caitlin, but there's a lot more we can do with the digital image analysis. And then there's some really complementary sort of protein type assays that, you know, sort of basic proteomic stuff. And I know Min asked a question about proteomics too, that we could do to get to that question, which is really important. One other thing, the other aspect of that I think is really important is, you know, while staining plaques tends to be rather mature beta amyloid fibrils, some of the oligomeric stuff, which people think is really the toxic component, isn't going to really show up in a typical plaque stain. Those can be measured with the solution phase assays. And so there's a lot of questions that can be asked about, you know, the different kinds of A beta and the different stages that it's in fibrillization, and as far as toxicity and burden in this setting as well.
Great. Let me go to another, actually the most recent question, just scanning the list of chats. Great talk. Did resilient subjects have any comorbid conditions, cardiovascular, diabetes?
So that was another, I remember we looked at that, and I don't think any of their health issues came out of that analysis either as being significant.
Okie doke. And questioning about measuring TDF-43 in the spinal fluid, CSF.
I think Jay-Z asked me about that once, and we had talked about doing that. So we're obviously doing, you know, there's CSF testing for amyloid and tau. I don't know where the field stands. I don't think anyone's doing it yet. And it's hard. I don't even know if there are any phospho-TDP antibodies, so you'd be looking at total TDP. I don't even know what that would mean, but
okay.
Yeah.
That's an area of tremendous interest and little success.
Yeah, the other is the imaging biomarkers, so having PET for that. But because I mean, you saw the inclusions for TDP, they're like these tiny little things, so I think that is far off as well.
Okay. Another question, Caitlin. Very nice talk, interesting studies. Have you or others performed proteomic and or RNA profilings in ACT brains? Do you know if CSF or blood samples were collected from the ACT cases?
So no CSF collected in ACT. The goal is to poke and prod these individuals as little as possible, I think, and that helps a lot with nutrition, or not losing patients. They're treated very well in the study. And I don't know about Dirk. Do they get blood? Or Eric, I saw was on here. I can't remember if ACT takes blood.
So they do have blood lately. I don't know that they have archival blood. I don't believe so.
And then, so the only CSF we would have is on some of them we collect it at autopsy. So that ventricular CSF.
We do have some blood stored in freezers, but it's not anything like a complete sample. It was done almost haphazardly at times, and if we do get this U19 award, we will have blood specimens on everybody.
Nice. And then I know that a number of ACT has had, they've done RNA-seq analysis in various brain regions. We did a study with the Allen Institute looking at a number of cases. So there are some with RNA-seq, and that's an area that I want to move into, is getting additional RNA-seq data on brains that are known to have this kind of overlapping tau and TDP-43 pathology as a future direction.
Okay. And here's a question that George actually wonders if Eric can answer that, since Eric is on. What are the major reasons for so many of the ACT subjects having refused autopsies?
Well, relative to what, George? Actually, it's quite a high rate for a community sample. If you condition your enrollment in the sample on accepting an autopsy, you get a very high rate of autopsy. But we actually have a higher rate than most, and we're one of the few people that have a lot of controls. So Caitlin had a 50-50 sample to work with, and we've had that for years. I don't want to be defensive about this, but part of the beauty of the ACT study is we know that the findings generalize to the general population. And people that have autopsies, we know, are a skewed population. Maybe back in Sweden in the 50s and 60s, everybody was getting autopsies, but it's a very rare event, really, in the United States nowadays.
Yeah, I think it's really a shame the degree to which there's been a spectacular decline in the frequency of autopsies generally, particularly in academic institutions. And I just wonder if, I guess people don't ask that question because it's difficult. I wonder what proportion of the no's are due to religious kinds of reasons or just other. I just don't have any information. I don't even know if there's literature on that.
Well, I think there is, but I don't know it well enough to respond to your question. But one of the things that one of our biostatisticians, Sebastian Henus, did probably, what, 10, 15 years ago, was he developed a method for us to actually, because we know the base population from which the people who agreed to autopsy came, you can tell if there are biases in the way the cohort was assembled from the base population. And if some of those biases might affect the results of your analysis, you can actually adjust for it. Or if the biases are uniformly distributed between those with and without an autopsy, you can ignore them. So we now, all the autopsies that are done in the neuropath group and all the papers that we write, unless they're very small sample sizes, we adjust for whatever a kind of bias might be in the group that's part of that analysis. And that's been accepted by the field, I think, pretty well. But you know, I think people you know, tend to have a lot of reasons for doing or not doing something, and a lot of it has to do with personal autonomy, in my view.
Just to add to that, George, it's not just a question of autopsy. It's a question of donating their brain for research, and so I think that adds some complexity to the question. I don't know that for a fact, and you know, so far the ACT cohort hasn't gotten into a lot of ethnic diversity. That's planned with the new U19, but then you're going to add a lot of cultural and religious barriers as you get more diverse as well. And so I have to echo what Eric said, though, as studies go where autopsy is not required for participation, the ACT study is doing really well, and they've got this inverse probability weighting protocol that Eric mentioned that really gets us back to the root of the population with the autopsy cohort that we have. So it's incredibly strong study, and we've been very, very fortunate to be able to work with it.
Another related question quickly is actually when I started the Alzheimer's Disease Research Center, I had a request that we get funding for full autopsies. And I had a salary slot for Ray Monat, a young pathologist. And the study section turned me down. They didn't think that was relevant. But one of the questions was, what's the effect of cardiovascular disease, for example, other diseases? And a full autopsy would be much more instructive. And so I was curious about, there's probably even a smaller percentage that agreed to a full autopsy, correct?
Well, I can answer that. So they're not asked. We don't have the funding or the expertise, to be honest, to be doing full autopsies on all these folks. But George, as the ACT study is envisioned for the future by Paul and Eric and others, you know, it really is getting to focus on life course more than ever before. And of course, life course has effects on the entire body, and the cardiovascular system is one of many organ systems where we would think it would have a, you know, real impact. And so as I think about this, I agree with you. We could learn so much by doing full autopsies. I just don't know how, at this point, that's something that we could do. We'd have to basically ask Desiree to help us with that.
Do I have time to add anything, or?
Please.
Okay. Yeah, this is Desiree. Yeah, so that is a really good question. And so we're trying to kind of do kind of bite-sized initiatives. So, sorry, my son's in the background. We would, you know, I think the way to start with that is with the rapid autopsy programs. And so, you know, with the recent, you know, developing interest in molecular characterization, and, you know, neoplasm is one area. So where we'll have, you know, known patients develop a protocol to do rapids, and then hopefully that'll be a platform where we can extend, where we can, you know, look at all the tissues of the body, including the brain, and you could, you know, be focused on certain diseases, certain neoplasms, or even just trying to understand normal human anatomy, physiology, the normal brain. So there's just a ton going on in this space, but we just haven't really been equipped or able to really do that, and it's going to start in these specialized areas, and then hopefully we can apply it and start bringing in more people to study this.
You know, and I would just add one thing. We have a whole bunch of trainees in our program and coming into our program through the Neuropath Fellowship, through the residency, and through the forensic fellowships that are really interested in this. And so there's an opportunity, I think, for Desiree's rapid autopsy program, and to potentially start to think about the brain, not just as the brain, but as part of the entire person, which sometimes we forget when in the neuropath core when we're just looking at brains. So, you know, maybe we should keep thinking about this with respect to our neurodegenerative disease cohorts, Desiree, because I think there could be a real opportunity with people who could take point and really propel this forward.
Terrific, building bridges. So this will be
our subjects and their families who they get a letter from us after every autopsy. So I think that the community is becoming more interested in the brain autopsy, and I'm guessing it would extend to the whole body autopsy.
We're just about through. The last question is TDP43 protein deposition really a discrete pathology, or should it be part of the AD pathology spectrum incorporating TIBRAC and TDP43?
Well, that was one of the things I actually asked myself when I started seeing this. Holy cow, this is just like the third protein of AD. And it did also make me really realize how different the earlier onset genetic forms are. We do see TDP sometimes in those, but it does seem to be at a much lower frequency than in the late onset. So I don't have a real answer for that, but and it gets complicated because TDP has been commandeered by the FTLD ALS folks, and so they want to think of this all as a spectrum of ALS and FTLD. So I think there's a lot of interest to really try to either separate or link it up with that, first of all, and some ways to do that would be diving into the proteomics and the structure of the protein potentially. But then as far as whether is it part of the AD disease or is it a concomitant pathology, I think that remains to be seen, and is one of the questions I hope some of this work will help us answer.
Super. So our hour's up. I wish we had an applause meter, Caitlin. This was a fantastic learning opportunity, and opportunity really for all of us not dealing with neurodegenerative diseases, Alzheimer's, to really learn more about, to get up-to-date knowledge. So how many months did we wait? The 6-month wait for you to do it. It's well worth it. So thank you so much, and thank you everyone for your questions. I know there are one or two that haven't been addressed, but maybe could address those directly to Caitlin.
Great.
So that completes this Pathology Grand Rounds, and we'll have another one in 2 weeks from now.
Great. Thank you.
So thank you all.
Great job, Caitlin. Thanks.